Welcome back, guys. Today we're going to be solving week 4 quiz for biology. Now the material, I've already covered it in my previous videos, so make sure to watch the previous videos again, especially part 2, the last maybe 10 minutes. It has the new material that you guys have, and I also posted another video which kind of refreshes your memory from previous material that you have taken before. So without further ado, let us begin. Which of the following accurately describe the effect of temperature on proteins? So remember, proteins have a specific structure, and their structure is what determines, or their shape is what determines their function. Now, if we really increase the temperature, we're going to be denaturing the protein. Now, so lots of protein shape will not affect function. That's not true. We said shape determines function, so that is not true. Above 50 degrees Celsius, the cross linkages that hold the protein together break. That is correct. That's why when we heat proteins for a very high temperature, they lose their shape. How do they lose their shape? Because their cross linkages break, and since they lost their shape, then that means their function is lost too. Which of the following are functions of bile? Remember, bile is produced by the liver and it is stored in the gallbladder, and both are accessory organs. Now, bile has bile salts. What does bile salts do? It helps emulsifies the fat, so it helps in the process of emulsification. And it's also a bit basic, as I mentioned before in my video, so it also neutralizes the acidic kind that comes from the stomach. How does the bacterium Vibrio cholerae cause the infectious disease cholera? So we said it infects the lumen of the small intestine, and it produces a toxin that causes the cells lining the intestinal wall to secrete chloride ions. Now, it causes water to move out of the lumen. That is incorrect. Remember, it causes water to move inside the lumen of the intestine. So we have to say which is true or false. In the colon, additional water is absorbed to transfer chyme into solid feces. That is true. The absorption of nutrients stops when the food reaches the large intestine. That is false. The bacterial flora of the large intestine can produce vitamin K. That is true. Feces are eliminated out of the rectum to the anus by peristaltic movements and muscle contraction. That is also true. The ileum is very long. Remember, we talked about the adaptations for the small intestine, because we said the small intestine is what is the main organ for absorption. So the ileum is very long, which allows plenty of time for food to be digested and absorbed. Its inner walls has millions of villi, which increase the surface area for absorption, and they have a very thin lining, as I mentioned before, to allow absorption to be quicker. Now, also true or false, hydrochloric acid in the stomach causes pepsinogen to be converted into its active form pepsin, and that is true. The acidic pH of the stomach is maintained by the saliva. Of course, it's not maintained by the saliva, it's maintained by the hydrochloric acid, so this is going to be false. And many bacteria and microorganisms taken within the food can be destroyed by the hydrochloric acid. That is true, because that was one of the functions of the hydrochloric acid. And just a reminder, hydrochloric acid has two main functions. It allows the stomach to have an acidic pH, which activates pepsinogen into pepsin. That's one of the functions. The other functions is it kills any bacteria and microorganisms taken in with the food. Circle the process that consists of the repetitive contraction and relaxation. Directly, whenever you see repetitive contraction and relaxation, it's going to be peristalsis. Okay, so sucrase, lipase, pepsin, salivary amylase, they're all digestive enzymes. So sucrase, remember, breaks down sucrose, and it's produced where? In the small intestine, okay? While lipase breaks down fats, and it's produced in the small intestine and in the pancreas. Now, pepsin breaks down. Remember, pepsin attacks proteins into smaller polypeptides, and salivary amylase breaks down starch in the mouth. Directly, you can know it's in the mouth because of the word salivary amylase. Cholera disease is caused by a pathogen that produces a toxin in the human body and leads to severe watery diarrhea and vomiting in infected individuals. It is usually observed in areas with poor treatment of wastewater and drinking water. Now, what type of pathogen? Remember, we said cholera is a bacteria, so it's a bacterium. Now we have to choose the order of steps of how it infects us. So of course, first of all, the bacteria is going to trigger cells lining the intestinal wall to secrete chloride ions. And then, after it secretes, the water potential in the lumen will become lower. Since the water potential is lower, water will move into the lumen of the intestine. And finally, large amounts of water and salts are lost in watery feces through diarrhea. Okay, so we've done this before, but let's do it again. So, of course, the first part is going to be the esophagus. And then we go from the esophagus to the stomach. And of course, the small intestine is what's inside, while the large intestine is what's outside. And which body system? Of course, it's going to be the digestive system. Okay? Let's go next. So drag and drop. Okay, so in the stomach, what enzyme do we have? We said we only have one digestive enzyme, which is the pepsin. And of course, it's going to be in an acidic pH. And what maintains the acidic pH? We said the hydrochloric acid. While in the small intestine, we have multiple enzymes. But here we're just looking at about the enzymes that break down proteins, because they said proteins are digested in the stomach and the small intestine. So trypsin is by the small intestine. And of course, it's going to be an alkaline, which means a basic pH. And what is the chemical that maintains the pH? We said it is sodium bicarbonate. Bile also plays a role, but the main factor, so pay attention for this, the main factor that plays the role is NaOH and not bile. And what are the nutrients after we digest proteins? Of course, we're going to get amino acids. And what happens to the excess of nutrients resulting from the digestion of proteins? Of course, we mentioned that in our first video in chapter 1. So they are broken down in the liver and they are changed into urea, which is excreted later on as urine. Okay, this is a very important table that you guys have to know. So in the mouth, what juices are secreted? Of course, we have the saliva. Do we have? Yep, saliva. Okay, so saliva is secreted, and we said what are the components of saliva? It has amylase, water, and mucus. So the enzyme that is present is going to be amylase. It breaks down starch into maltose. Now in the stomach, we have the enzyme pepsin, breaks down proteins into polypeptides. Now what is the juices secreted by the pancreas? Of course, it's going to be pancreatic juice. And we have three main enzymes that are secreted: lipase, trypsin, and amylase. But we have to look at the substrate so we know where to put them. Okay, so of course, we're going to put trypsin here. It breaks down proteins into polypeptides, while lipids or lipase is what breaks down emulsified fats into fatty acids and glycerol. And finally, we have amylase, which breaks down starch into maltose. And finally, we have the enzymes in the small intestine. They're made by the intestinal glands or released by intestinal glands. And some examples are lactase, sucrase, and maltase. So of course, maltase is what breaks down maltose into glucose. Lactase breaks down lactose into glucose and galactose. And finally, sucrase breaks down sucrose into fructose and glucose. So where's sucrase? Sucrase is right there. Perfect. So we have to match each enzyme to its substrate. Of course, lipase breaks down fats, amylase breaks down starch, trypsin breaks down proteins. And remember, all those three are released by the which organ? Of course, it's going to be the pancreas, which is an accessory organ. Okay, we've done that before. So the function of the esophagus transports food from the mouth to the stomach. In the stomach, it continues the chemical and mechanical digestion of food. Now, the big organ is the liver. What does the liver do? Liver produces bile, and it is stored in the gallbladder, which is right under the liver, and the pancreas is over here. It secretes trypsin, lipase, and amylase into the small intestine. Oral rehydration therapy is used to restore water balance in the case of course, diarrhea, because diarrhea is the loss of water. It suggests the administration of a dilute solution of sugars and salts. I've mentioned that before in my video of part 2, so make sure to watch it for you to understand everything we're doing right now. So what is a condition that involves the discharge of watery feces due to reduced water absorption in the large intestine? Of course, it's going to be diarrhea, and make sure you guys know how to spell it. Okay, perfect. Underline the enzyme that produces glucose and fructose and circle its substrate. So the enzymes that produces glucose and fructose. So glucose and fructose make up sucrose. So that's the substrate. And the enzyme is going to be, of course, sucrase. Underline the intestinal enzyme that breaks down lactose. Of course, what breaks down lactose is going to be lactase. And what is lactase made up of? It's made up of glucose and galactose. What ensures the breakdown of maltose? Of course, maltase, and maltose, as we said before, is made up of 2 glucose molecules. Emulsification, remember we said it's a form of mechanical digestion. During this process, bile salts break large lipid globules into smaller droplets, increasing the surface area for the action of lipase. The enzyme in the gastric glands, whenever we're talking about the gastric gland, it's going to be in the stomach. So what enzyme do we have in the stomach? Pepsin. It's released in its inactive form called pepsinogen, and as food enters, what stimulates the conversion? Remember we said hydrochloric acid. Match each component of saliva to its role. So amylase, of course, ensures chemical digestion, water acts as the solvent, and mucus sticks the food particles together. Okay, so what does the liver do? We said the liver removes excess glucose. Do we have an option glucose? Yep, we do. From the blood and stores it in the form of, remember, in mammals, it's going to be in the form of glycogen for short-term use. And then the liver removes the nitrogen group or the amino group from excess amino acids. Remember we said what was the process called? Deamination. We mentioned that in our first video. And many proteins found in the blood are made in the liver, for example fibrinogen, which is used in blood clotting. And the liver also produces bile salts. The liver also stores iron, and the liver stores fat-soluble vitamins. Remember, 2 fat-soluble ones were A and D. Bile contains, what does bile contain? We said bile contains, of course, bile salts. So bile contains bile salts, and the process is known, we said, as emulsification. And also pay attention for the typing. I don't want you guys getting mistakes for typing. And this has increased, we said, the surface area of the lipids, which allows lipase to act more quickly, which is the digestive enzyme. Okay, what is this diagram? Of course, this diagram represents a villus, which is found in the small intestine, and that's the main for absorption. What is structure X? Of course, the outermost layer is going to be the epithelium. Structure Y is going to be the blood capillaries, where sugars and amino acids are absorbed, while structure Z is the lacteal, and mainly fats are absorbed inside here. Okay, in order to allow plenty of time for digested food, the ileum has to be, as we said before, it has to be very long. Now the ileum finger-like projections, now the answer should be villi, but you're gonna get it wrong if you put villi. They put cilia, but I'm 95% sure it should be villi, and increases the surface area. Each villus is made up of cells that have small numerous projections on the surface known as microvilli. This further increases the surface area. Each villus contains, remember, for absorption of fat, it's going to be a lacteal, while for amino acids, glycerol, and some fats are blood capillaries. And finally, the lining, we said, has to be very thin. So the inner wall has millions of villi and, of course, microvilli. Pay attention again for the spelling to increase the surface area. So increase the surface area for absorption. It's highly supplied with blood capillaries, okay? So blood capillaries. and of course we said a lacteal to allow the movement of fats. Absorption of food takes place across the villi in the small intestine, in the small ileum, in the ileum mainly. Here we have to be a bit more specific. They're telling us in each villus there's a dense of blood capillaries and a single lymph vessel. Glucose and amino acids are absorbed into the blood capillaries. Some fatty acids also pass through the capillaries and not arteries, and some fats move from the intestinal cells into the lacteals, which are, of course, lymphatic capillaries. These lacteals carry a fluid called lymph that will rejoin the blood circulation, and most of the water is absorbed in the small intestine. Okay, some water is absorbed in the large intestine, but most of it is in the small intestine. Okay, so I posted also another video for those concepts, so make sure to check it out. What is the name of test used to verify the presence of fats? Of course, it's going to be ethanol emulsion test, and if we get a positive result, which means presence of fats, we're going to get a white opaque emulsion. Starch, glycogen, and cellulose are three important biomolecules. Which class? Of course, it's going to be carbohydrates. What is the type? Now, they're made up of multiple glucose molecules, so they are polysaccharides. Which monomer? We said it already, which is glucose. Storing in plants, of course, it's starch. Storing in animals is glycogen, and providing support is cellulose. Okay, so fructose is what is a monosaccharide, so the one sugar. Another example is glucose. Now, disaccharides like maltose have a sweet taste and are soluble in water. Another example could be sucrose, which is made up of glucose and fructose, while polysaccharides are insoluble in water and do not have a sweet taste. An example would be glycogen and starch. Starch with a polysaccharide does not react with Benedict's solution because it's not a reducing sugar. So with the test we use iodine test. A positive result is indicated by a presence of a blue color. Oh, my bad. Proteins are large biological molecules. They contain carbon, hydrogen, nitrogen, and oxygen, and sometimes sulfur and phosphorus. The basic units are amino acids, and they are joined by peptide bonds. Okay? Peptide bonds. Okay, so for protein, it's going to be a bioret and gives a purple color if protein is present. Nucleic acids are made up of nucleotides, and they're composed of a simple sugar, a phosphate group, and a nitrogenous base. And what is the shape of the DNA? We say the shape of the DNA is called a double helix. Okay? So let's double check. It's going to be a double, let me type it down, a double helix. Double helix. And remember guys, A pairs with T and G pairs with C in our DNA. That's the question. Adenine pairs with thymine. Also guys, watch out for the spelling. Guanine always makes a base with cytosine. What is composed of one glucose and one fructose? Of course, it's going to be sucrose. And what is the bond that joins them? We call it a glycosidic bond. Remember, ester bond is in fatty acids, peptide bond is in proteins, which joins two amino acids. When two molecules of a glucose linked by a glycosidic, we're going to get a maltose. Disaccharide in milk, of course, it's going to be lactose, and one glucose and galactose also bound by a glycosidic bond. Lipids are used as a source of energy, and fat always insulates the body. Lipids are essential for the formation of cell structures such as the cell membrane, and some steroids function as hormones. An example would be testosterone. Okay, how do we test for reducing sugar? We add Benedict's solution, heat above 80. It's going to change from blue to cloudy to green to yellow brown and then to brick red. And solution remains, of course, is going to remain blue if no reducing sugar is present. Okay, so what is this molecule? We have 3 fatty acids, we have a glycerol, of course it's going to be a triglyceride. A represents the ester bond, B is our glycerol, and C are our fatty acids. How many water molecules? Remember, since to form a bond we lose a water molecule, so we formed 3 ester bonds, so we're going to lose 3 water molecules. What is a common test for vitamin C? DCPIP, and of course an indication would be that it becomes, I'm not sure, I think it's going to lose its color, so it's going to become colorless. Enzymes released by the intestinal gland, of course, what do we have? So sucrase acts on sucrose and it's made up of glucose and fructose. Now we have peptidase, which works on polypeptides and releases amino acids. Now we have maltose acts on maltose, which releases glucose. And finally, we have lactase, which acts on lactose, which breaks down to glucose and fructose. Okay, now we have to order the events. So of course, first of all, the bacteria is going to infect us. After it infects our small intestine, it's going to release a toxin, which causes the release of chloride ions. Now that will change the water potential. After we change the water potential, water will move, and then that's how the final result would be the loss of water. Okay, so name this disease. As you guys can see from the figure, it's a small intestine, and what infects our small intestine we said is cholera. So this disease is cholera. And what happens as a result of this disease? We said the bacterium produces a toxin, cells lining secrete chloride, and the water potential in the lumen increases. Nope, it decreases. That's why water moves in. Water moves out. No, as we just said now, water moves in, and large amounts of salts and water are lost in the feces. That's it for this video. Now they're gonna get us one mistake, but I'm 90% sure it's going to be, that's the correct answer. What did we do wrong here? Oh, okay, so we spelled it wrong. So guys, watch out in your exam. You don't do the same mistake I did and spell something wrong. Okay? And that's it. That's it for today's video. Thank you guys for watching. Make sure to leave a like, comment, and subscribe. If you have any questions, let me know. And best of luck on your exam. Hopefully, we're going to see a lot of hundreds again this week. Thank you guys, and best of luck.